Afternoon or good evening, depending on where you are in the world, and welcome to another day of MDDI's Focus on Fundamentals. It's a master class on reusable medical device cleaning, disinfection, and sterilization validations. This 3-day webinar series is sponsored by Nelson Labs and broadcast by Informa. And this is day 2. Today we'll be discussing disinfection validations. Now, my name is Peter Krass. I'll be your moderator today. And before we begin, I have a few very quick announcements. First of all, this webinar is designed to be interactive. There's a dock of widgets at the bottom of your screen, and you can use that to learn about today's speaker, download resources, participate in the Q&A session that we'll have toward the end of the presentation. The slides will advance automatically throughout the event. No need to click. Also, please be sure to do click on the Register Now button. It's in your viewing window. This will allow you to join us for the remaining Focus on Fundamentals class tomorrow, when we'll be discussing sterilization validations. And finally, if you have any technical issues during this, technical issues during this webinar, please click the Help widget. It's at the bottom of your screen. Or you can type your issue right into the Q&A area. And either way, we'll be glad to offer you one-on-one assistance. Let's move on to our presentation for day 2. How should disinfection validations be performed for reusable medical devices? Discussing today's topic, we have Griffin Kamek, expert technical consultant at Nelson Labs. To learn more about Griffin, please visit the Bios widget. And with that, I'm going to hand it over to Griffin Kamek. Griffin?
Hi, and thank you for that introduction. As mentioned, my name is Griffin Kamek, and I am a technical consultant at Nelson Laboratories. So what I do at Nelson Laboratories is I help medical device manufacturers with their reusable or their reprocessing validations for their reusable medical devices. Meaning I help with test method justifications, master product determinations, things like that. And today I will be talking about disinfection validations, as mentioned, for reusable medical devices and what that means and how should they be done. And I am very excited to talk about this topic. I love disinfection validations. My time as a lab analyst, this was some of my favorite testing. And so I am excited to talk about this topic and to help you understand a little bit more about what is required for your reusable medical device. So as just a reminder for what a reusable medical device is, and this may be just kind of a review of what was discussed yesterday if you attended the presentation yesterday. But a reusable medical device is a device that is intended for repeated use on different patients with appropriate decontamination and other processing between uses. Reusable medical devices can range from invasive to non-invasive and to even non-patient contacting devices. So it's a large array of devices that fall under the definition of a reusable medical device. What we have pictured here are just some examples. So we have surgical devices, we have blood pressure cuffs, and even monitors. So those, again, those non-patient contacting devices that are sometimes viewed more as capital equipment. Those are reusable medical devices, and as such need validation work for their cleaning and disinfection procedures. Additionally, it's also worth noting that reusable medical devices for single patient use are also under this definition for a reusable medical device. So devices such as menstrual cups or CPAP masks, those are things that are considered reusable medical devices, even though they are for single patient use only. And so a lot of what we will talk about today will apply to, again, those devices for multiple patients, but also those devices for single patient use. So in the process overview for a reusable medical device, and again, this might be just a little bit of a review from yesterday, but just to go over the process of what is done on a reusable device, it'll be used in whatever procedure or whatever it's designed to be used, after which it may go through a point of use processing. So this is just an initial treatment to remove or prevent the drying of soil or contamination on the device. This is usually done at the bedside, in the hospital room, and it's just really meant to, again, avoid any contamination from drying on that device. After which it'll go through a thorough cleaning procedure. That's what was discussed on the presentation yesterday, was how to do the cleaning, how to do the validation for that thorough cleaning procedure. After that cleaning procedure, the device will then either go through sterilization or disinfection as the terminal step. And so today, what I have highlighted here is the disinfection portion, and that's what we'll be talking about. And so as a reusable device, one option for the terminal step is disinfection, and then within that option, there are actually 3 disinfection levels that you can choose from for your device. That is low, intermediate, or high level disinfection. And I will talk about what that means, what each of those disinfection levels mean, and we'll also go over how to know what disinfection level is appropriate for maybe your particular medical device. So you might be asking yourself, what is really the difference between cleaning and disinfection? Why do I have to do both validations? Are they not the same process? And that's an excellent question. And the FDA guidance document that was initially published in 2015 and revised in 2017 gives a pretty great definition for each of those processes. So for cleaning, cleaning is the physical removal of soil and contaminants. The methods and agents used for cleaning should be designed to remove soil and contamination effectively. Effective cleaning should minimize the soil transfer from one patient to another or between uses in a single patient. Should prevent accumulation of residual soil throughout the product's use life and allow for successful subsequent disinfection and sterilization steps. So that is what the intent for cleaning is. The intent, as it says here, is just to remove soil and contaminants. Whereas for disinfection, that process is intended to kill microorganisms. And the methods and agents used for disinfection should be designed to achieve appropriate microbicidal effects. So that's just a great definition for the 2 processes, why they're different, and why we have to typically do the validation separately, because the intent of each process is clearly different. So we need to show that each process can adequately do what it's intended, what it is intended to achieve. So why do disinfections? Why are they an option? Well, disinfections must be performed if the device is unable to be sterilized. In a perfect world, it would be great if every device could be sterilized. That would ensure that all devices are safe to be used. However, that's just not the case. Some devices are just unable to be sterilized, whether that is due to the size or the material made of the device. And in some cases, it's just not necessary because the device has low risk of transmission of infection. Additionally, disinfections may be applicable for facilities where sterilization is not available. So for some of those devices that may be sterilized, but disinfection is the minimum requirement, maybe where you are marketing your device, some of the facilities don't have access to sterilization methods, and so disinfection is a great option for those facilities. And then lastly, in some regions, maybe disinfection is a interim requirement for the processing of your device. Maybe where you're marketing, you need to show that the device can be adequately disinfected even before sterilization, which is what we see in Europe. We see a lot of countries in Europe wanting to see thermal disinfection data before sterilization. And we'll talk a little bit more about that a little bit later in the presentation. So there are a few different kinds of disinfection methods out there. It's the different methods of disinfection are essentially broken down into two categories, chemical and thermal. Within the chemical category, you have manual chemical disinfections. These are performed by soaking the device or spraying or wiping the device with a chemical disinfectant. You also have automated chemical disinfections. This is where automated equipment is used to disinfect the device, such as an automated endoscope reprocessor. The other method is thermal, which again, you have manual and automated thermal disinfections. And for automated, those thermal disinfections would be done in a typical washer disinfector that you might find in a hospital. And then a manual thermal disinfection would be something like pasteurization or boiling water. And that's kind of reserved more for those devices that are used at home. So again, some of those devices that may be single patient use, but are still reusable. So again, another example of that would be like a menstrual cup. They would go through pasteurization, thermal disinfection, or put into boiling water. So as I mentioned, there are 3 disinfection levels, and it could be hard to know what disinfection level you may need, but there is a great guide that you can go by. Essentially, the disinfection level for your device is going to be based on how your device is classified according to the Spalding classification, which is what I have in the table here. The Spalding classification is based on the device's patient contact. So a device that will only contact intact skin or maybe non-patient contacting, that is classified as a non-critical device, and as such requires low-level disinfection or intermediate-level disinfection. Examples of that would be a blood pressure cuff like I have here, or a monitor, a surgical light in maybe an OR room. That would be considered a non-critical device. And really any other piece of capital equipment, carts, IV poles, those are all considered non-critical devices and require either a low-level disinfection or an intermediate-level disinfection. The next classification is semi-critical. These are devices that contact mucous membranes or non-intact skin. And these devices at minimum require high-level disinfection. So these are devices such as the speculum or a endoscope, or some other examples would be a transesophageal probe or a transrectal, transvaginal probes. Those are all semi-critical devices. And at minimum, they need a high-level disinfection. And then lastly, the last classification is critical devices. And those are devices that contact sterile areas of the body, including blood pathway contact. And these devices require sterilization. So for critical devices, there is no minimum disinfection level they have to meet. They have to be sterilized. And these are devices that are going to be used in surgical procedures where it's important that they are sterile during their use. So additionally, for thermal disinfections, there are broken into high-level, low-level, and intermediate-level disinfections as well, based again on that Spalding classification or patient contact. In the EU, they prefer something called A Subzero testing, or A Subzero values, which we'll discuss what that is a little bit later in the presentation. And those are broken down as listed here. So for your invasive devices that will be terminally sterilized, you'll need an A Subzero value of 600, and then your non-critical devices just at least an A Subzero value of 60. The U.S. doesn't recognize the A Subzero value, so they still rely on the definitions for high level, intermediate level, and low level disinfections, even for thermal disinfections. So we'll talk about what those A Subzero values are, and what that means a little bit later in the presentation, and how to perform those sorts of validations. So as you noticed, for non-critical devices, it has two disinfection levels that you can choose from. You can either have a low-level disinfection or an intermediate level disinfection, and you may be asking yourself, well, how do I know what level of disinfection my device needs? The guidance document, AAMI TIR 12, which was just recently updated in 2020, well, about 3 years ago, gives some pretty great guidance on how to know when to do a low level or an intermediate level disinfection, which I've kind of broken down into this table here. So if your device is going to be patient contacting or maybe contact healthcare personnel, and the likely contamination will be blood or maybe other potentially infectious materials or other bodily fluids, then your minimum disinfection level should be intermediate level disinfection. If your device is going to contact intact skin or maybe be non-patient contacting, and the expected contamination is not blood or is not bodily fluids or otherly potential infectious materials, then your device at minimum needs a low level disinfection. And then if your device is non-patient contacting and it is not expected to see blood or otherly potential infectious materials, then maybe your device just needs to be visibly clean. And that is in line with some verbiage in the FDA guidance document that I referenced earlier, that from 2015. It does say that there are some instruments that will likely not become contaminated with pathogens, and as such may only need a thorough cleaning and not disinfection or sterilization. That is that's in line with what it says in that FDA guidance document. However, the conservative approach is just to do a disinfection validation even on those devices that are not likely to see blood or other potential infectious materials, just to ensure minimize that risk of cross-contamination. So this is just what it says in that AAMI TIR 12 document in Annex F. This is just the guidance it gives for knowing when to choose an intermediate level disinfection or a low level disinfection. And just kind of a quick summary, based on the table that we just went over. And what's written here is essentially if your non-critical device is going to contact intact skin and be blood contacting, or we'll see bodily fluids, then you should do an intermediate level disinfection. Whereas if your device is just going to see intact skin or no contact and not see any blood, then a low level disinfection would be appropriate for your device. And it's important to choose the right level, because that'll affect how the validations are performed. It'll affect how stringent the requirements are for your device. So you want to make sure you choose the right disinfection level for your particular device. So again, just to go over, critical devices need sterilization, and what that means is you need to have you need to show a sterility assurance level of 10 to the -6 or greater. And for semi-critical devices, at minimum, you need a high-level disinfection. And what that means is you need to show a 6-log reduction of an appropriate mycobacterium species on your device using your specified disinfection procedure. And then for non-critical devices, the disinfection will be either low level or intermediate level. And for a low level disinfection, you just need to show a 6-log reduction of 4 vegetative organisms. And then for an intermediate level disinfection, you need to show a 6-log reduction of 4 vegetative organisms. And in addition, you need to show a 3-log reduction of an appropriate mycobacterium species. Those 4 vegetative organisms are the same 4 used during a low-level disinfection. So a low-level disinfection and an intermediate level disinfection, the criteria are very similar, but there is just that added organism, that added mycobacterium species at a 3-log reduction for that intermediate that just kind of increases the testing requirements. So now that you have assessed your device and you are ready to put together your disinfection validation, you know what disinfection level you need. You will then need to research what organisms, what microorganisms do you need to challenge for your disinfection level. And that's some of that's what this table lists here. So this table just covers the different microorganisms used for the different disinfection levels and where those are referenced. For high-level disinfections, for chemical disinfections, mTera is typically what's used, and that's found in AMI TIR 12. If you're doing a thermal disinfection, a thermal high-level disinfection, then you're going to want to look at using a thermophilic mycobacterium species. There's not really much guidance on what organism to use, so you'll want to make sure you research prior to testing and ensure that you choose an appropriate thermophilic mycobacterium species to really challenge that thermal disinfection process. And then for low-level disinfections, these are the 4 vegetative organisms that are commonly used: S. aureus E. coli, P. aeruginosa, and K. pneumoniae. And these are referenced in that AMI TIR 12 document, as well as the CDRH Class 2 Special Control guidance documents for washer disinfectors. They are listed in there as well. I will say, if you have not reviewed AMI TIR 12, I would highly recommend that as you prepare for any disinfection validation, that you review that document. It has a lot of great guidance in there and can help you understand what really your device needs. So the reason why we use these particular devices just really comes down to their resistance to chemical and physical activity. So that's what this diagram is showing. It's showing the most resistant organisms on top to the least resistant organisms on the bottom to chemical and physical activity. In addition, I've added kind of where the sterilization and disinfections fall in line in relation to the microorganisms, as well as the device classification: critical, semi-critical, and non-critical. This information is found in AMI TIR 12 in Annex F. It's not presented in this way. It's presented through a table, but I found this to be a little bit more visually appealing and a little bit easier to read. So I've switched into this format. And so once you've chosen your disinfection level, once you know what organisms you'll be evaluating for your disinfection level validation, now you're ready to do your disinfection validation for your medical device. So there are essentially 3 test phases for a disinfection validation. You have your neutralization testing, your microbial reduction evaluation, and then if your device is patient contacting, you're going to want to do some sort of test to evaluate for disinfection residuals or disinfectant residuals. And that's typically done through cytotoxicity testing. But again, that's only for patient contacting devices. So if your device is a non-critical, non-patient contacting device, you don't need to worry about that test phase. So for microbial reduction, there are the microbial reduction phase of testing. That is where we're actually evaluating how effective the disinfection procedure is in relation to your medical device. And that is broken down into 3 steps. First being inoculating the devices or the samples with the microbacteria, or the microorganisms with the bacteria, followed by taking those samples through the disinfection procedure, whether that be automated or manual, chemical or thermal methods. After the disinfection process, we will extract, or you will extract the samples, and then you will test for microbial reduction. So you'll look for a recovery from the test samples, recovery of microorganisms from the test samples, and compare those to a positive control and calculate a log reduction value. For the sample size and controls, for a disinfection validation, you will have 1 positive control. That's going to be 1 sample that is inoculated with the microorganisms, but it is not processed through the disinfection procedure. And this will give us kind of a starting microorganism level on the device, and it's what we will use to calculate the log reduction for the test articles. For test articles, typically there are 3 that uses 3 samples or 3 test articles, and those are samples that will be inoculated, and they will be then processed through that disinfection procedure. AMI, the new update to AMI TIR 12 from 2020 does not define a sample size like it used to. However, it is still typical to do 3 test articles. With that in mind, because AMI TIR 12 does not define a sample size, you may want to assess, the complexity of your device when determining how many test articles to do. So if your device is a little bit more complex, you may want to do maybe more than 3 devices. And if your device is maybe a little bit more critical, you may want to do more than 3 devices. If you have an endoscope or a probe, or something that is a little bit more difficult to disinfect, 3 test articles may not be sufficient. For the neutralization test, that will be 1 sample, and it will only be processed through the disinfection procedure. And then the cytotoxicity testing or the disinfectant residual testing, that's typically done with either 1 to 3 samples. And again, those are samples that are processed through the disinfection procedure, but are not inoculated. Now, what is that neutralization test that I mentioned? The neutralization test is essentially a method suitability test. It is ensuring that that we are neutralizing any disinfectant left behind after the extraction procedure. And what that means is, after the disinfection process, you need to extract your devices using a neutralizer, some a solution that has properties that are known to neutralize the active ingredients in whatever disinfectant that you are using, so that we can accurately recover any viable organisms that may be left on the samples after that disinfection procedure. The reason why this is so important is because any disinfectant residuals that may be left on the device can inhibit growth during the incubation of the microorganisms, which can impact the test article counts and would essentially give false data and an inaccurate final result. And it might show that your device is safe to use when perhaps it is not safe to use. So that's why it is very important to do. The criteria for neutralization testing comes from USP 1227. and you just need to show a recovery of 50 to 200% when the test plates are compared to the control plates. So once you have shown effective neutralization, you are ready to move into the microbial reduction evaluation phase of your disinfection validation. And the first step of that, if you recall, was inoculating the samples with the appropriate microorganisms. And this is done through direct inoculation. So you will directly inoculate only those clinically relevant areas and those areas that are hard to disinfect with the chosen microorganisms based on your disinfection level. For inoculation, there is really only one test soil. So unlike the cleaning validation, the cleaning validation from yesterday's presentation, if you attended that, there were a lot of different test soils that you can use during your cleaning validation. There's a lot of choices out there. Whereas for disinfection, it's the opposite. There's really just one choice, and it's going to be a 5% bovine serum. So you'll inoculate that 5% bovine serum with your microorganisms and then place that onto your medical device. If you're testing multiple microorganisms, so if you're doing a low level or intermediate level disinfection, you're going to be evaluating more than one microorganism. And you're going to want to decide if you're going to do mixed suspensions or if you're going to do individual suspensions for each organism. AAMI TIR12 does recommend doing individual suspensions just to limit the impact of competitive inhibition between the different microorganisms during a mixed suspension. So that is kind of the safe option and the more conservative option. If you do perform the inoculation using a mixed suspension, you may just want to have you just want to ensure that you are taking into account any competitive inhibition and ensuring that that is not occurring during your testing. So after the inoculation, once the devices are inoculated, you'll then, as a part of the validation, move into the disinfection portion, where you will take your device through the disinfection procedure using whatever procedure you have in your instructions for use, which will be based on your disinfection level. So for high-level disinfections, these procedures for chemical disinfections are usually performed through soaking the device, so fully immersing the device into that high-level disinfectant and allowing it to soak for a specified time. That time is gonna range from 12 minutes to anywhere from 12 minutes to 45 minutes to 90 minutes. It really depends on what high-level disinfectant you choose, which is important to know that if you're going to be marketing in the US, you want to make sure that you choose a FDA-approved high-level disinfectant. So you can follow the link that I have posted here, and that will take you to a list of FDA-approved high-level disinfectants. And you want to choose one of those disinfectants. High-level disinfectants, they're going to be peracetic acid solutions, hydrogen peroxide solutions, or glutaraldehyde solutions, or similar solutions as those. And so taking into account the material compatibility with these different solutions is something you want to consider when you are preparing or when you're doing your disinfection validation. So there are some challenges that you'll run into that you may see when performing a disinfection validation. And one of those challenges is that high-level disinfectants their label claims are evaluated or are tested using coupons, which is what I have pictured here. So there's, the coupon is what's being challenged. And that is how that specific contact time or soaking time is determined for that high-level disinfectant? Well, reusable medical devices are a lot more complex than a coupon. They have challenging features, design features, such as crevices, areas that actuate lumens, that might challenge the disinfectant a little bit more than that coupon. And so it is not uncommon to see disinfection procedures for medical devices that extend past that extend past the label claim contact time for that high-level disinfectant. And it really just comes down to the fact that the medical device is more complex than the material used to evaluate the disinfectant. So that was for high-level disinfectants or disinfections. So if you are doing a low-level or intermediate-level disinfection procedure, you're going to choose a low-level or intermediate-level disinfectant, and these are products that are commonly pre-saturated wipes that you'll wipe your device with. Occasionally, these are products that are sprays that you'll spray onto your device. There are some high-level, low-level intermediate or low-level disinfectants that require you to soak the device, but that is pretty rare. And you want to make sure that your device is EPA registered as well. And these chemicals, if you're marketing in the US, you want to make sure that they are EPA registered. And these chemicals are isopropyl alcohol, quaternary ammonium compounds, hydrogen peroxide, sodium hypochlorite, bleach. Those are some pretty common active ingredients for low and intermediate level disinfectants. So again, there are challenges with low level and intermediate level disinfections. Low level, these disinfectants, like high level disinfectants, are their label claims are tested using coupons, which again, these are a picture of some commonly used coupons. They are either non-porous or maybe just slightly porous materials, and they don't have a lot of features on them. But again, we are applying this disinfectant to a more complex device, such as this a blood pressure cuff or a monitor, or something that's going to have crevices and buttons that may be difficult to disinfect. So again, we see often that the contact time listed on these low level or intermediate level disinfectants just not be sufficient to achieve the needed disinfection level. So going back to your disinfection validation, once you've inoculated your device and then you've taken it through your disinfection procedure, the next process that you'll do is you'll extract your device. You'll extract the device to recover any viable microorganisms that may be left on there after the disinfection procedure. Extraction methods that are commonly used are sonication, manual shaking, orbital shaking, flushing for lumen devices, or swabbing. You may recognize this slide from yesterday's presentation as well, because a lot of the same methods used for cleaning validation, extractions, you can apply for disinfection level extractions as well. You just want to make sure that you choose an appropriate method for the device based on its size and delicacy. And again, you want to make sure you choose an appropriate neutralizer to ensure you're neutralizing any residual disinfectant. And then once you've extracted, you'll then plate the extract fluid onto agar plates and allow those to incubate and then enumerate. This is just a picture of some of the organisms used for low level and intermediate level, just placed on just various types of agar. Just so you can get an idea of what those organisms look like, and how fun it can be to count. And, they're pretty interesting. They have some pretty interesting interactions with the agar sometimes, and so it makes them pretty bright and fluorescent, which is kind of fun. So that, once you've enumerated your test articles, as I said, you'll then compare those counts to your positive control. and then you'll calculate a log reduction. And then you just need to ensure that your log reduction meets whatever the requirements are for your particular disinfection level. Now, some things to note between the US and the EU is that the US prefers more glutaraldehyde-based solutions or similar solutions. So products such as Metricide or Cydex OPA, those are used predominantly here in the US. Whereas outside the US, we see that they prefer more peracetic acid and hydrogen peroxide solutions. So what that means for your disinfection validation is if you're looking to market in each region, you're going to be doing maybe more than one disinfection validation based on where you're marketing and what disinfectant you're using. It's very difficult to make an equivalency claim between two different disinfectants. Even disinfectants that have the same active ingredient, it is difficult to make equivalency claims. So if you're going to list more than one disinfectant in your IFU, then more than likely you're going to need to show data to support each disinfectant listed, and that disinfectant is able to be properly used on your particular device. And then also between the US and the EU, we see that thermal disinfection validations are performed a little bit differently. So thermal disinfection validations in the US are performed using microbial methods, meaning we will take ampules, just little glass ampules, and fill those with the organism, attach those to the device, and then take it through the thermal disinfection procedure, or we will just directly inoculate the device and take that through the thermal disinfection procedure. And then we will look for any microbial recovery after that process. However, in the EU, they prefer more A Subzero methods. So this is a temperature-based method, very similar to the F Subzero method for sterilization. It's a time versus temperature kill rate using a specific Z value. It uses thermocouples, so it does not use microorganisms. It uses thermocouples that you'll attach to your device and monitor the temperature over the thermal disinfection phase. So again, if you're marketing in different regions, you may want to take into account how, where you're marketing and how that thermal disinfection is being performed. So one thing I do want to mention is ISO 17664-2 was updated in 2021, and it clarified some items or some requirements for non-critical devices. So it states that at least one validation method shall be specified for each applicable stage of processing of the medical device. And then it goes on to say disinfection may be carried out concurrently with cleaning of the medical device. And then note 2 further says the requirements for cleaning and disinfection are stated as separate clauses in this document. However, when the steps are concurrent, the requirements for both stages can be considered as one. So essentially what this means is, oftentimes cleaning, for non-critical devices, the product you're going to use for cleaning is the same product you're going to use for disinfection. So you can do validation work to show that both processes are effective in one validation. So you can do a combination cleaning and disinfection validation. And that, again, that's in ISO 17664-2, which is specifically for non-critical devices. And then the last thing that I want to talk about is just disinfection as an interim process. So it is not uncommon to see disinfection done on critical devices that are going to be sterilized anyway. And TIR 12 does address this, again, in Annex F. It does say that disinfection can be done as an interim process to ensure that the device is safe to handle during the preparation for sterilization. So after it's been cleaned and while it's being wrapped, you can do disinfection to ensure that the device is safe to handle. That is typically done for washer or for thermal disinfections. We see that pretty commonly. And we see that as a requirement coming from a lot of countries in the EU. So we see interim disinfection procedures being validated and being requested for to ensure that, those sterile processing technicians in the sterile processing department are safe when they handle the devices. They've been observed to prepare the devices for sterilization, not wearing gloves. So just to mitigate any transmission of pathogens, we see countries in the EU requesting interim disinfection procedures on those critical devices. And we typically see it more with thermal disinfections or thermal processes. So this is just some common thermal disinfection parameters you'll see coming from Europe. And they're really just based on different kind of what country you're marketing will kind of will determine what thermal disinfection you have in your IFU. And then last, I talked a little bit about A Subzero earlier, and I just want to go over that again. This is a definition for A Subzero from a few standards and guidance documents. And again, it's very similar to the F Subzero for sterilization. So it's a time versus temperature kill rate that uses a specific Z value to ensure that to calculate a Subzero value. So what you'll do is you'll attach thermocouples to hard to reach locations or worst case locations on your device, place that into the washer disinfector, program the thermal disinfection stage. Often that is 90 degrees for 1 minute. That's what we see is kind of the most common. You'll then monitor the temperature over 5-second intervals. And then using that data, you'll calculate an A Subzero value. For most devices, an A Subzero value of 600 is what's required. We do see some requests for 3,000 for some of those devices where disinfection is the terminal step. So that just brings me to the end of my presentation. I appreciate everyone joining this. The last few slides are just reference standards for reprocessing validations in general. So I'd invite you just to review those. There's a lot out there. So there's a lot out there. So I just invite you to review them and become familiar with them. If you have any questions, just don't ever hesitate to reach out. And that just brings me to the end of my presentation. And I, again, appreciate everyone joining and coming out today.
Okay, and this is your moderator, Peter. We're going to go to Q&A in just a moment. But before we do, I'd like to mention that we have a webinar survey. We'd like everyone to take that. It's available on the right of your presentation window. If you've closed the survey, you can reopen the widget. Just click the icon along the bottom of your screen. And thank you in advance for filling out the feedback form. Your participation in this survey will help us to serve you better in the future. All right, let's move on to our question and answer portion. You can still ask Griffin a question. Just type it into the text box that's located to the right of the presentation window, or you can click the Q&A icon at the bottom of the screen. We have lots of questions, and we'll try to get to all of them. But if we're unable to, we'll share the questions that we didn't get with Griffin, and he can reply to you offline. All right, let us go to the first question. Boy, there's a lot of them, and they're good ones. Okay. For a non-critical device that's intended for both home and hospital use, skin contact would typically be the hands, as the device is intended to be used in contact with clothing and not directly on the patient's skin. Here's the question. How then would I make the determination between a low-level disinfection and an intermediate level?
Yeah, that is an excellent question. So it is a device that will most likely just come in contact with non-gloved hands, if that's correct. in which case you just want to assess, the intended use of the device. Is it likely to become contaminated with blood or other bodily fluids? And if the answer to that question is yes, then you may want to do an intermediate level disinfection. If the likely contamination is no, then a low level disinfection would be appropriate. Now, I just to clarify, or just to further expound on that, rather, the key point, or the key question to ask yourself is the likely contamination. You don't necessarily want to think about worst case, because you can really kind of dig yourself into a hole and really do some testing that is just not needed for your device. So you really want to look at what your device is intended to be used for and what is the likely contamination, and then make your determination based off of that.
Okay, let's see what our next question would be. Just give me a second. Oh, right, here's a question. You had mentioned earlier, Griffin, EPA guidelines, and the question is, does my Class 1 device need to be EPA registered?
Class 1 device? For the
right.
device? No, so it would be more for the chemical disinfectant that you are applying, or that you're going to use for your medical device. You want to make sure that the chemical disinfectant for your low or intermediate level process is EPA registered. Again, that, specifically for the US only. So yeah, that's just more in relation to the disinfecting chemical.
Okay. Here's another question. During the disinfection procedure, what neutralizer is used? What neutralizer?
So there are a lot of different neutralizers out there. And it really just depends on what the active ingredient is. So you want to make sure that whatever neutralizer you're using has some properties, a chemical or an agent in it that is going to neutralize whatever the active ingredient out there. So, or whatever the active ingredient is for your disinfection or disinfectant. So there's not really one neutralizer, one magical neutralizer for everything. We wish there was, here at Nelson Labs, because that would make it very easy. But there are different neutralizers, and it really, again, just comes down to what your disinfectant is. And that's something that I can answer offline and give some a little bit more information about kind of what some of those common active ingredients are.
Okay, great, thanks. Here's our next question. Is it acceptable to do a single device in triplicate to achieve the equivalent of 3 samples?
Yeah, that's an excellent question. So yeah, that is acceptable. The more important part is just achieving at least 3 data points. Typically, we do that with 3 samples because that makes the testing go the smoothest, and it makes things go faster. But if not in every case are you going to have that many samples available for testing. And so we've done that in the past. So yeah, you can do 1 sample and just use it multiple times for your different, for whatever sample size you're needing.
Great. All right. Here's the next question. You mentioned, hold on a second. Here we go. You mentioned disinfection as an intermediate step to protect handlers, but is this step simply suggested, or is it actually required by the standard?
Yeah, that is also an excellent question. And I might say that after every question, I guess, but it is not a requirement per any standard. So there's no written requirement that says you have to do a disinfection as an interim process before sterilization. What we've seen feedback from hospitals in Europe that have requested for data on these interim processes. Again, typically that's more for thermal disinfection processes and washer disinfectors, but we've seen feedback from countries, from notified bodies and hospitals in the EU specifically that are requesting this data before accepting the sample, or the device rather. In the U.S., it is not a requirement. It is not an expectation or a requirement to have this process in your instructions. If you do put an interim process or an interim disinfection process in your instructions, you might be asked for validation data. If your device is patient contacting and you have an interim chemical disinfection process, you may want to just do a biological risk assessment on that chemical you're using during that process. Even though that's not the terminal process, you just want to make sure that that chemical isn't going to affect by way of biocompatibility. So that's just something to consider as well. But again, for the US, those interim processes are not required.
Okay, good. Next question is about how you categorize devices. So here's the question. If you have an invasive critical device like a retractor, and then a second instrument mounted on top of it, and the second one does not touch the patient, how would you categorize the second instrument? Is it semi-critical or critical?
Yeah, that is a great question. I would technically, if you're following the strict Spalding classification, that is technically a non-patient contacting device. However, as which it'll be a non-critical device, right? However, with its use with the retractor and where it's being used, we often just tend to treat those as critical devices, and then additionally, a lot of those devices, they're often packaged in sterilization trays with those critical devices, so they're all treated as the same. So something, again, if you recall from yesterday, if you attended the presentation yesterday, Alpha spoke on master products or worst case devices. So for trays that are, these surgical trays or these instruments that have, similar use and similar materials, you can group them into product families and then choose a worst case device or a master product. And that's what you'll do your validation on. And so often those devices that attach to, in your example, attach to the retractor, although it may be non-critical, it does somewhat get lumped into its critical counterpart or its critical device that it attaches to, and is treated as a critical device.
Okay, thanks, Griffin. Looks like we have time for one more question. And here it is. Is mixed inoculation method better than individual?
Yeah, so great question. And again, I guess these are all great questions. I don't need to say that every time, but as I mentioned in my presentation, AAMI TIR12 does recommend that you use separate suspensions during your disinfection validation. And the reason for it is to ensure that there is no competitive inhibition with your microorganisms. So if you're looking at AAMI TIR12 and you're following that, AAMI TIR12 would say it's better to do separate suspensions because you're limiting that competitive inhibition or that potential for that. If you can show that competitive inhibition is not occurring, then mixed suspensions are fine to do. It's not necessarily better. It's, I would say it would save you a little bit of time because that would be, you would be applying 2 organisms at a time rather than 1 at a time. But it's not, I wouldn't say it's necessarily better, but you do just want to make sure that you are showing or that you can defend that there is no competitive inhibition occurring.
Okay, well, we're coming up on the top of the hour, so we did not get to all the questions, so we will share them with Griffin, and he can reply to you offline. So, Griffin, before we say goodbye, any closing statements?
No, I just want to say thank you to everyone that joined today, and I am excited to see all your questions, and I will get to them as soon as I can.
All right, thanks, Griffin. We appreciate your time and expertise today. I'd like to thank our sponsor, Nelson Labs, as well as everyone out there in the audience. We appreciate your attention and participation. Within the next few hours, you'll receive a personalized follow-up email. It will contain details and a link to today's presentation on demand. And if you have colleagues and peers who couldn't join us today, please invite them to listen to this event on demand. This webinar is copyright 2023 by Informa. The presentation materials are owned by or copyrighted by Informa Markets, and the individual speakers are solely responsible for their content and opinions. On behalf of our guest speaker, Griffin Kamek, I'm Peter Krause. Thanks, and we look forward to seeing you again tomorrow. Thanks.